The YouTube stream is live now. Everyone should be able to find it on the main FWP YouTube page.
Just let everyone know as you start tuning in that we're just giving folks a few minutes. It's not quite 6:30, so we'll be another 5 minutes or so, and then we'll get going.
Izzy just stated that the video is live now. Should show up on the webpage.
Great. So again, as folks are tuning in, we're going to give ourselves another 2 or 3 minutes here just to make sure people have a chance to join, and then we'll get going. If you've just tuned in, we're going to start the meeting here in a few minutes, just making sure everybody has a chance to jump on here from the beginning. All right, I'm gonna slowly get us rolling here so we can try to stay on time this evening. My name is Vivica Krauser. I'm the Information and Education Manager here in Region 2 for Fish, Wildlife, and Parks. And first, I just wanted to say thanks for probably for many of you tuning back in tonight. We, as you know, had a technology issue last week, and so we're sorry to tie up another evening of your time, but we're glad we could get it turned around pretty quickly here and have it working tonight. So welcome and thank you for being here again. I'm going to quickly turn it over here, but I just wanted to go through a couple minor little logistics. So on the page where you are watching this right now, on the page where the EA for this project is also posted, right underneath the video is call-in information. And the only time you need to use that number is when we get later on in the agenda this evening, when there's a time for questions and comment. If you want to ask a question or make a comment, that's where you need that link. Otherwise, everything else is streaming right here for you to watch and tune into in that way. And we will also post a recording of tonight's meeting, so you can tune back in if there's pieces you miss or want to watch again. We'll have that up later this week. So, but that number's right there, and you should be able to call that at the time. And then I will be monitoring those numbers as they come in and unmuting folks to make questions or to make comments and ask questions. There is also another, underneath that information, there's a place to make comments at any time just through the web portal. So people have been doing that already, and the comment deadline has been extended. So because of our delay last week from the original meeting, we are extending the comment deadline through August 14th. So hopefully that gives everybody just a bit more time. So that's noted on the webpage too, but I wanted to make sure to point that out. And with that, I'm going to turn it over to Patrick, who's our biologist that is the lead of this project.
Thanks, Vivica. Well, good evening, everybody, and thanks for taking the time to join us this evening. I'm Patrick Youthy. I'm the Blackfoot Area Fisheries Biologist out of Missoula with Fish, Wildlife, and Parks, and I'll be working alongside our team members on this project to oversee implementation and then also conduct the post-project monitoring. Tonight, we're going to go through a presentation, kind of walk everybody through a lot of the content that's in the EA and distill it down to a lot of the highlights that are probably most relevant and of most interest to a lot of you tuning in tonight. Before I do, I want to take a minute to introduce a number of our interagency team members. There's a diverse amount of expertise on tonight's panel, and everybody will be available for questions. So I'd like to start off by introducing Randy Arnold, who is our Regional Supervisor with Montana Fish, Wildlife, and Parks here in Region 2 in Missoula.
Hi, thanks, Patrick.
Next, I'd like to introduce Pat Saffel. He's our Fisheries Program Manager with Fish, Wildlife, and Parks here in Region 2 and works out of the Missoula headquarters office.
Thanks, Patrick. Yeah, I'm the Fisheries Program Manager. I've been working on this project for many years and have visited the area many times. I think I've been in most far reaches of the area and yeah, overseeing the EA process and supervising the project. And Patrick, thanks.
Next, I'd like to introduce Don Scar. He's a bureau chief out of our Helena headquarters office with Fish, Wildlife, and Parks. And he's also a national expert on the use of piscicide and frequently teaches the American Fisheries Society piscicide application course.
Good evening, Patrick. Thank you. Glad to be here.
And next we have Pat Clancy. He's a retired FWP fisheries biologist who throughout his career implemented a number of piscicide projects. And since retirement, he has been gracious enough to contract on and assist with a number of piscicide projects throughout the state, including this proposed project.
Thanks, Patrick.
And next we have Josh Lattin. Josh works for the Forest Service on the Helena, Lewis and Clark National Forest, and he is a wilderness manager on the Lincoln Ranger District and has been assisting us with development of this project, as well as assisting with all of the the Forest Service administration associated with conducting this project in the wilderness.
Howdy, folks. I'm happy to answer any questions that come up in regards to wilderness character or anything specific to the Scapegoat Wilderness. Thanks.
Well, I think with that, we'll dive right into the presentation. And yeah, at the end, there'll be ample time for any questions for any of us. And then we'll move into a separate period to take any comments you might have towards the project in general or the EA specifically. So West Slope cutthroat trout are the most broadly distributed of the 14 cutthroat trout subspecies that we have in the Western US. And in addition to being Montana's state fish, it is also a fish with significant cultural, social, as well as economic value. And historically, it was broadly distributed in Montana in the upper Missouri drainage, as well as all of the drainages on the western side of the divide. And this particular map is showing in gray those areas that used to be historically occupied, where they have since been extirpated from. And if we look at our genetically unaltered, so our pure West Slope cutthroat trout populations, they occupy just over 10% of this historic range. So a really significant decline. And this has been attributed to habitat degradation, historical overharvest, as well as the introduction of non-native species, in particular rainbow trout, which readily hybridize with cutthroat trout in water bodies where they co-occur. And a more recent and emerging threat, as well as one that's anticipated to pose one of the greatest challenges to long-term persistence, is climate change and the associated changes in abiotic and biotic conditions in the water bodies where they occur. So from a conservation and management perspective, we've implemented harvest restrictions in water bodies where West Slope cutthroat trout naturally reproduce. We've also implemented a number of habitat restoration projects to address known limiting factors. And then a fairly common, as well as probably one of the more effective long-term conservation techniques, has been the intentional isolation of pure populations, either taking advantage of an existing pure population and erecting a barrier to prevent the spread of invasives into that area, or locating drainages that already have a man-made or natural barrier and removing non-natives and establishing pure cutthroat populations. So we look up to the South Fork of the Flathead drainage up near Kalispell, above Hungry Horse Dam. That area has been managed as a pure West Slope cutthroat trout project, and they applied a lot of rotenone to some headwater drainages where hybrids were present in order to achieve those conservation benefits above the dam. In Silverbow Creek, there was an existing pure population, and a specific fisheries barrier was implemented to isolate that population. And the West Fork Bitterroot, Painted Rock Dam isolated pure cutthroat above it. And actually, Region 2 Fisheries just finished up last year with a 3-year rotenone treatment in a small tributary above Painted Rocks Dam to deal with one of the known sources of hybrids in that drainage. And then looking down to Southwest Montana in the upper Madison drainage, the site of Cherry Creek, which you'll hear more about later in this talk, but it has a lot of similarities to this proposed North Fork project, and the fact that it has a natural barrier with a large expanse of habitat above it, in which non-natives as well as hybrid cutthroat trout were present. They went in, applied rotenone over a number of years, removed all of those hybrid and non-native fish, and then established a pure conservation population of cutthroat trout. And then in yellow, I've highlighted the North Fork project area to put it in some context. And while there is precedent for this type of action, I want to highlight that it's somewhat rare that all the building blocks really align to have a project of this size be very successful. And the fact that we have that in the North Fork project area, which I'll get into here momentarily, I think it's really incumbent upon us from a conservation standpoint to really seize this unique opportunity. So moving into the North Fork project area, at the bottom, we have North Fork Falls, which is a 50-foot natural fish barrier. Above it, we have a large expanse of really high quality, complex cold habitat. There's 19 perennial streams with about 85 miles of stream habitat. And this project area is located entirely within the Scapegoat Wilderness. So one of the reasons why it still has that high quality, intact habitat without any sort of anthropogenic habitat degradation. And as such, it's likely to remain that way for a very long time. So I want to briefly walk through the project timeline. This project, as Pat had mentioned earlier, it's been a long time in the making. So going back to the early 2000s, initially it started out as kind of just backcountry fisheries investigations, trying to get a handle on what is up there and how broadly fish are distributed. So it started out in the lakes and then morphed into electrofishing surveys throughout the tributaries and main stem sections of the North Fork River. And then more recently, we've been using environmental DNA to not only corroborate those electrofishing results, but also refine our known fish presence model. And throughout this time, we've deployed temperature loggers to look at overall thermal suitability in the project area, as well as help to inform some modeling exercises to project what temperatures might look like well into the future. And as I mentioned, a number of these similar projects have been completed around the state. So around 2013, 2014, with that in mind, that's really when this current project was kind of conceptualized, and Fish, Wildlife and Parks started pursuing funding in 2014 to hopefully implement a project such as this. And then the Forest Service secured some additional funding in 2016 to help with implementation. And so since that time, our interagency team has been working to really hone in on the proposed scope of action, as well as develop an implementation plan. And over the last year and a half, we've really refined the MIPA and NEPA processes, and what we're left with is the current proposal that we've put out to you and that we're discussing tonight. So a quick summary of all the types of information, baseline data that's been collected. In addition to all the fisheries information, we've done extensive surveys to look at amphibian presence, as well as get a handle on what aquatic insects are present. And not only to just understand what's up there, but also as part of our statewide piscicide policy, we need to have pre-treatment information on those organisms to compare with after a piscicide treatment to make sure that all of our mitigation measures were successful and that we didn't have any unintended consequences. So we know that fish stocking occurred extensively above North Fork Falls. And here I've labeled on the map all the various specific stocking locations that have been referenced in a lot of these historic stocking documents, whether it was from newspaper articles from the 20s or our FWP stocking database. And a lot of these early records mention undifferentiated cutthroat trout, which we now know were actually Yellowstone cutthroat trout, so not native to the Blackfoot drainage. Rainbow trout were also widely stocked, and then some pure West Slope cutthroat trout were also stocked in the drainage. This map is showing all of the electrofishing sites, and you can see we've covered a large extent of the drainage, and the yellow triangles demonstrate the sites where we captured fish, and then those red triangles are the sites where we did not capture fish during electrofishing surveys. And at these sites, as well as in the lakes, we collected genetic samples from fish to get a handle on species presence as well as hybridization status. And so from those results, we distilled down to knowing that fish are currently present in 19 of the tributaries above the falls, and then 3 of the 4 lakes. East Twin Lake, which is not pictured here, had been stocked in the past, but due to likely winter kills or maybe even summer kills, it no longer supports fish. It hasn't been recolonized since one of those events likely occurred. So we do have 3 lakes that currently have fish. And putting the genetic results in the context of the entire Blackfoot watershed. So this is showing the last 20 or so years of genetic surveys throughout the entire Blackfoot Basin. And it's kind of a busy figure. So what I really want to highlight are these closed red circles. These represent sites where pure West Slope cutthroat trout were sampled. And then the combination of either fully black circles or black and green or black and yellow represent rainbows and various hybrid forms of rainbow, West Slope, and or Yellowstone cutthroat trout. So those non-native hybrid fish that we've been discussing. And what you can see here is that the middle and upper Blackfoot watershed are our current strongholds for pure West Slope cutthroat trout. And within the North Fork drainage in particular, we do see some hybridization, but all in all, it retains a high degree of genetic purity for the West Slope cutthroat trout below the falls, as well as has pockets throughout the North Fork drainage with pure West Slope cutthroat trout. When we move into the project area above North Fork Falls, we see it sticks out like a sore thumb. We've got hybrid fish with very little West Slope cutthroat trout admixture, ranging from 0% to about a max of 17% West Slope cutthroat trout contribution. So basically a hybrid swarm that's predominantly rainbow and Yellowstone cutthroat trout. I do want to briefly mention this. You see this red dot up here in the upper North Fork. It's still unclear whether that fish was just an error, but this particular pure West Slope cutthroat trout, one lone fish from Cooney Creek, could not be replicated. So we're chalking that up to some sort of analytical error or just a mishap. And when we remove that data point, again, the cutthroat trout or West Slope cutthroat trout composition ranges from 0 to 17% throughout the drainage or throughout the project area, excuse me. In addition to being an important cutthroat trout stream, the North Fork Blackfoot also supports the most robust migratory population of bull trout in the Blackfoot, as well as within the middle and upper Clark Fork drainages. So extremely important. And they currently spawn and rear right up to the North Fork Falls. Now, bull trout, they're a really good canary in the coal mine. Out of all of our trout species, they have the lowest thermal tolerance. So they're a really good indicator for any potential consequences from stream warming. And the slide I'm showing right now is from a modeling exercise from the Forest Service that shows predicted bull trout spawning and rearing areas from 1980, as well as under anticipated climate scenarios around 2040. And 2 things I want to highlight. You see this significant contraction from historical to predicted future suitable habitats. Even this, our robust stronghold area within the lower North Fork is anticipated to likely be too warm for bull trout come mid-century. Whereas we look above North Fork Falls and we see a large proportion that's anticipated to be highly suitable even under warming scenarios. Now, I don't have the same slide for West Slope cutthroat trout, but it follows the exact same pattern in terms of a contraction from present to future distribution. But West Slope cutthroat trout have a slightly warmer thermal tolerance compared to bull trout. So their mid-century predicted distribution will be a little bit broader than this of bull trout. But at any rate, a large proportion of the proposed project area is anticipated to be highly suitable, even when lower areas in the Blackfoot drainage are going to be too warm. And I want to mention at this point, if you've looked at the EA, you've probably seen the mention of bull trout. And if you tuned into our bioassay back in 2018, there was a lot of discussion of bull trout being included in the project. I just want to mention now that the current project proposal is only dealing with rotenone application, as well as the establishment of West Slope cutthroat trout. You know, our Fish, Wildlife and Parks kind of grand vision for the North Fork Native Fish Conservation Project is just that, a whole native species assemblage within the project area. But bull trout will be addressed through a separate and future environmental analysis, due to we still have a lot of planning to do surrounding that action, whereas the cutthroat component is really refined and ready to go, and is directly tied to the rotenone application. So that is the reason for only considering the cutthroat trout conservation component of the project at this time. Looking at fish population status, we also see a pretty stark difference in terms of fish density within the project area compared to very similar adjacent drainages within, say, Montour Creek and then the Landers Fork. We see that these hybrid fish are present at much lower densities compared to pure West Slope cutthroat trout in these neighboring similar drainages. And that's led to this running hypothesis that these hybrid fish are poorly adapted to this environment. And if we had pure West Slope cutthroat trout population, they'd likely be present at higher densities using this habitat more to its full potential and reference condition, as well as likely being more broadly distributed. So more recently, to further inform all of these electrofishing results, DNA sampling, we've been using environmental DNA to look at upstream extent of fish within tributaries, as well as confirm the presence of other fish barriers within tributaries. So giving us a known point on how far upstream fish occur within a given water body. And all of this information over the last 15 or plus years has been distilled into this current proposed action, which is to apply rotenone to about 67 miles of stream, as well as those 3 fish-bearing lakes. And ultimately, we have 2 primary objectives with this. One is to establish a conservation population of cutthroat trout in the project area. And by that, I mean a population that has greater than 90% genetic purity. This conservation criteria came out of the West Slope, or I guess the statewide cutthroat trout conservation MOU, and we place high conservation value on populations that either have are genetically unaltered or have minimal hybridization, less than 10%. So that's the goal we're shooting for. And then furthermore, the second objective is to reduce or substantially reduce or eliminate these hybrid fish above the falls that further pose hybridization spreading risk downstream of the falls in the lower North Fork, as well as other areas of the Blackfoot drainage. And with that, I will turn it over to Pat Clancy, who will provide some background on rotenone, as well as some of the details about how we plan to implement the rotenone treatment.
Okay, thank you, Patrick. Can you hear me okay? Hello, can you hear me okay?
Yeah.
Yes.
Yes.
Okay. So Patrick, you're going to have to advance the slides, I guess, as I proceed through here.
Okay.
Because you have the control of that, right? I don't.
I do. Yeah. I'll listen.
Okay, so yeah, I've been asked to discuss what rotenone is and how we use it. How does it work in these projects? Rotenone is a naturally occurring compound that is present in plants and stems of tropical and subtropical plants from pretty much around the globe, circum-equator. Go ahead, Patrick. It's been used by native people in South America and Oceania for hundreds of years to capture fish for food consumption. And to do this, they they harvest the plant and they either pulverize it and scatter the pulverized plant into the water, or they actually, in some cases, I guess, chewed it to make a paste, which they applied to their body, and then they swam in the water. And as fish became disoriented and easier to catch, they were harvested for consumption. And I've read something recently that this was documented still occurring in as late as 2009. The chemical formula of rotenone is carbon, hydrogen, and oxygen, so a pretty simple chemical formula. Okay, Patrick. For our purposes, we can use it as either a liquid or a powder. The powder is just simply the plant material ground up very finely into kind of a dust. The liquid, which is what we primarily use, is the rotenone is extracted from the plant and then mixed with, go ahead, Patrick, mixed with some other detergents and surfactants that help distribute the rotenone in the water and help it improve solubility so it distributes through the water well. And at this point, I think we want to make sure and point out that on page 35 of the environmental assessment, there's a discussion of some of these inactive ingredients that are in the formulation of rotenone. But we've learned that the formulation has changed, and specifically that the compound that's discussed on page 35 of the EA, it's called phenidipho 99, is no longer a component of the liquid rotenone formulation that we would be using on this project. It was removed because in certain conditions, it made the rotenone real kind of viscous and gummy, and it hard to flow out of the device that we apply it to the stream in. So talking with the company, they were able to modify their formula and remove that particular compound. For our projects, we use it in pretty low concentrations, never more than 4 parts per million, and typically not even as high as 3 parts per million. The compound we would use is 5% active ingredient. So 5% of the product is rotenone. The other 95% are these inactive detergents and surfactants. So if we were to apply rotenone at a concentration of 4 parts per million of the liquid formula, that would be 200 parts per billion of rotenone. We did some bioassays up in the North Fork of the Blackfoot a couple of years ago to help us determine how much rotenone we would need and how frequently along a stream course we would need to apply it. Its effectiveness is influenced by the chemistry of the water you apply it to, by the temperature of the water. Sunlight and aeration both degrade it at an accelerated rate, and then organic material in the water binds it up and makes it ineffective. Okay, Patrick. The fish absorb the rotenone through their gills into the bloodstream, Patrick, and it's then distributed throughout the body by the circulatory system and absorbed into the individual cells of the body. And once in the cell, the rotenone interferes with a chemical process that produces energy for the cell. So the cell cannot produce energy and then just slowly dies and becomes non-functional. Rotenone is not harmful to humans or terrestrial animals at the concentrations we would use it in this project. And you can read about on pages 33 to 37, you can read about the impacts of human health, terrestrial animals, and non-target organisms. Okay. So to determine how much rotenone we would apply at any given stream in this project, we do a number of things. We actually measure the discharge of the stream, how much water is flowing down the stream in cubic feet per second, which is the standard expression of how to express the volume of water in a stream. And then we apply a non-toxic green dye to the stream at the uppermost point where we think we would apply a rotenone. And we follow that leading edge of that dye plume downstream, and every 30 minutes, we mark where that leading edge of the dye plume is. And we periodically have to add more dye to keep that leading edge of that plume visible. And then once we've determined, we've measured the 30-minute intervals down the stream, we can then conduct these bioassays. We conduct 2 different types of bioassays. One is called a serial dilution bioassay, and the other is a persistence bioassay, or what I call a travel time bioassay. Patrick. So we did these, as Patrick, I think, mentioned, in 2018 in 3 different tributaries of the East Fork of the North Fork, Scotty Creek, Blondie Creek, and the East Fork of Meadow Creek. Over on the left-hand side, you can see the discharges of each of the three streams. Blondie was 0.15 CFS, Scotty Creek 1.3, and the East Fork of Meadow Creek 5 cubic feet per second of water flowing down the stream. So three pretty different volumes of water. Notably, Blondie and Scotty Creeks had high organic loads, a lot of decaying vegetation in those streams. Okay, Patrick. And we noticed that generally throughout the East Fork of the North Fork, the streams tended to look like they had more organics in them than those streams over in the main North Fork side. So we would expect the rotenone to perform better on the main North Fork side than in the East Fork of the North Fork. So we're doing these bioassays in locations where we expect the rotenone to perform not at its peak. Okay, go ahead, Pat. I'm not going to get into the nuts and bolts of how we do these bioassays, but this serial dilution bioassay we use to determine what the lowest concentration of rotenone formula in the water is that will give us 100% mortality of test fish. And so this figure right here is depicting several different containers with a mixture of stream water and different quantities of rotenone in them. And there's fish in each bucket. And at one of those concentrations, it's going to kill all the fish in it, and some may not. And then go ahead, Patrick. And then the other bioassay we do is this one I call a travel time bioassay. And on this one, we use those 30-minute intervals that we've identified going downstream, and we place fish in flow-through containers. Then we apply rotenone at a point upstream, and we determine how far downstream that rotenone carries and still provides 100% mortality of the fish in these containers. So typically we see at least 90 to 120 minutes of carry of the rotenone that provides 100% mortality of the sentinel fish. We have seen greater in some instances. Both of these tests we conduct for 4 hours, because that's how we do when we apply the rotenone during a project, we design it around a 4-hour application of rotenone. Okay, go ahead, Patrick. So the serial dilution bioassay, the one that tests the lowest concentration that will kill 100% of test fish, you can see there the different concentrations that we tested. The minimum concentration that achieved 100% mortality was the second highest one of 0.25 parts per million of rotenone formula. And that's a little bit higher than what I've seen other places. In other projects we've conducted this, done this bioassay, we've seen the test fish die with, at some of the lowest concentrations we've ever tested. Okay, Patrick. This slide is a little busy, but this is showing the results of that travel time bioassay. And in the far left-hand column, you can see the 3 streams and their stream discharge. The second column is the concentration of rotenone that we applied to those streams. So Scotty and Blondie each got 1 part per million of the rotenone formula, and the East Fork of Meadow Creek got 2.75 parts per million of rotenone formula. And we, again, Blondie and Scotty have high organic load. The East Fork of Meadow Creek is what you might envision as a classic mountain stream. It's pretty clear water, gravelly bottom, not much organics. So we would expect the rotenone to perform better there than in Blondie or Scotty. Okay, Patrick. So you can see that the numbers in this table where that red box is and in all those other cells show the results, what happened to the fish at our 30, 60, 90 minute sites and so on. So the first number indicates the number of fish that were unaffected. The first number in each cell indicates the number of fish that were unaffected by the treatment at the end of the test. The second number is the number of fish that are distressed but still alive. And the third number is the number of fish that were killed during the test. And so you can see Blondie and Scotty Creeks, we had all 3 test fish at each location get killed at the 30-minute site, but none below that. And in fact, on Scotty Creek, which is a higher discharge, so the rotenone tends to carry better in higher discharge streams, we did see that the 2 fish at the 60-minute site below our application point were distressed at the end of the test, but the next morning they had fully recovered. Okay, Patrick. As you might have noticed by now, on the East Fork of Meadow Creek, which I said has a much lower organic load, and we applied a higher concentration of rotenone, we saw 100% mortality of sentinel fish out to 3.5 hours of stream flow time below where we applied the rotenone. And Pat, go ahead, Pat. And we had 60% mortality at 4 hours. Now that's a very good return. So gather those two bioassays together will help us determine how much rotenone to apply at different locations within the project area and how far apart to space our application points. And it's good to know this information because we don't want to space our rotenone application points so close together that we get somewhat of a snowballing of the rotenone. We also don't want to space our application points too far apart so that we have undertreated sections of stream. So when it comes to actually conducting these projects on the ground, this is an illustration or a photo from a different project, that Cherry Creek project that Patrick mentioned, where we're applying. We've gone through our pre-treatment work. We've measured the stream discharges. We've done our dye testing so we know how every half-hour interval on the stream. And then from the information we get in our bioassays, we can determine how frequently, which specific points along the stream we want to apply the rotenone. So in this photograph, the white bucket with the green hose going down to that bowl over the stream is applying a mixture of stream water and rotenone to the stream at a very slow rate. That net bag labeled sentinel fish has 3 or 4 fish in it. They are not affected by the rotenone being applied by the bucket in this photograph. They are measuring the rotenone coming down from upstream. So each day we would have a series of these points along the stream where we would apply rotenone, and we would have sentinel fish near each of those to measure the results or the effect of the rotenone from upstream. We would also have additional sentinel fish posted well downstream of where we end each day, so that we can kind of monitor. Go ahead, Patrick. We can kind of monitor then how far each day's treatment carries, and it kind of serves as a double check on our bioassays. We can make adjustments if we aren't getting good carry. If, for instance, in this photograph, that those sentinel fish were not seeming to be affected by the treatment, we would set up an intermediate station between the one shown here and the next one upstream and run it for probably a couple of hours to try and fill the gap. Okay, go ahead, Pat. So this is just a shot of the mixture of stream water and rotenone. As you can see, it's just a tiny little trickle coming out of the bottom of that bowl where these are automatic dog watering bowls. So we just drilled a tiny hole in the bottom of them, and then there's a float there that regulates how much liquid is in the bowl at any given time. And that gives us a very steady application of rotenone to this. Go ahead, Pat. Patrick mentioned there are 3 lakes in the project area. Those 3 lakes are information on them is in the table at the bottom of this slide. On Parker Lake, it's big enough that we felt we needed to have 2 boats with motors to apply the rotenone to be able to get that done in a timely fashion and an effective manner. On Lower Twin and Meadow Lakes, we think we can do those fairly easily with just 1 boat that's powered by oars instead of a motor. You can see also the volume of the water in those lakes. So if we want to treat those lakes each at 1 part per million, then that third column shows you how much rotenone we would apply to each lake to achieve 1 part per million. And so to do this, there's 2 people in the raft. One of them guides the raft in a systematic grid pattern up and down and around the lake. The second person just kind of keeps the yellow container you see in the raft there. They keep that filled with water and rotenone, and then that little electric pump on top of the tank is powered by the battery, and it just runs, it just pumps the rotenone out into a nozzle behind the boat. And then as we go around the lake in that systematic grid pattern, the rotenone gets dispersed around the lake. Okay, Patrick. Rotenone can be neutralized with a chemical called potassium permanganate. There are 2 ways to do that. The potassium permanganate, you can see the chemical formula is just potassium, manganese, and oxygen. When we get it, it comes in containers, and it's just a granular, almost like a sandy consistency. We can either apply it as that dry granular formula in those volumetric feeders you see on the left photograph, or we can mix it with water and apply it as a liquid like you see in the right side photograph. And in both instances, it turns the water purple, and then that purple color fades to a light brown as the potassium permanganate neutralizes the rotenone. I'll point out here that on that photo on the left, you see the two different volumetric feeders. The Fish, Wildlife, and Parks policy requires that we have two independently operable neutralization systems on each of these projects. So if something were to go wrong with one of them, we have a completely independent system that we can fall back on. So once we apply this potassium permanganate to the stream water, we can fully neutralize the rotenone within 30 minutes of contact time. Go ahead, Patrick. But there's a bit of a trick to it. The organisms in the stream have a demand for some of the potassium permanganate. So while we're applying the potassium permanganate, some of it is not available to neutralize rotenone because different organisms in the stream are consuming it. So in order for us to make sure we're neutralizing the rotenone, we have to determine what that organic demand is. And we also then have to determine how much of the potassium permanganate we need to neutralize the rotenone. And then we give ourselves a little bit more than that so that we can measure the concentration of this potassium permanganate at that 30-minute site below the neutralization station. We have a meter that we take a water sample, put it in this meter, and that meter spits a number out at us, and it tells us what the concentration of potassium permanganate is at that 30-minute site. So if we see a surplus, and we try to have a surplus of about 0.5 to 1 part per million at that 30-minute site, if we see that surplus, that tells us that we are neutralizing the rotenone and meeting that organic demand. So that we, it's just a kind of a double check to make sure that we're, neutralizing the rotenone. Go ahead, Pat. So the neutralization stations on the North Fork of the Blackfoot would be at least 30 minutes of stream flow time above the waterfall. So we would be neutralizing the rotenone before it goes over the waterfall. And just to make sure that we don't impact bull trout or any fisheries downstream of the waterfall. Okay, Pat. So you see a red circle just came up on the left-hand photograph. There's an orange bucket in that circle. That bucket has holes drilled in it and there are fish in there. So as the rotenone comes downstream to the neutralization site, it's going to kill the fish in that bucket. And that's what we want. We want to be able to kill fish all the way down to the neutralization point, and then we start neutralizing the rotenone. We cannot stop neutralizing the rotenone until a subsequent batch of fish that we put in that bucket lives for 4 hours without any distress. So, the initial group of 4 or 5 fish in that bucket dies. It's going to take probably 4 to 6 hours at least before we would put another batch of fish in there to start measuring whether they're still being affected by the rotenone. And we cannot turn off the neutralization until a batch of fish that we put in there survives for 4 hours with no distress. Okay, Patrick. So in order to determine how much of the potassium permanganate is consumed by the organisms in the stream, we did another bioassay while we were up in the East Fork in 2018. And to do that, we applied 3 parts per million of potassium permanganate for a little over 4 hours. There was no rotenone in the water during this test. We're simply measuring how much biological demand there is for the potassium permanganate. So about half an hour before the end of that 4-hour test, we took a measurement at the site 30 minutes downstream from where we were applying the potassium permanganate. And we found that there was 0.7 parts per million potassium permanganate remaining in the water. So that indicates that 2.3 parts per million were being consumed by the stream. So that gives, go ahead, Patrick. That gives us the information we need to determine, to help us determine what the concentration of potassium permanganate we will need to apply to neutralize the rotenone during the actual treatment. I guess just a little bit more about potassium permanganate. I skipped over something, but it's used in municipal water treatment plants to remove metals and potentially harmful bacteria. And it also can be used early in the disinfection process of water treatment plants to cut down on the amount of chlorinated products they have to use later to purify the water. So to remove a little bit of the mystery, hopefully, about what potassium permanganate is, it is used in municipal water treatment plants for purification of water. Thanks, Patrick.
Thanks for that, Pat. So Pat really dove down into the nuts and bolts behind rotenone and how we plan to use it in this project. I kind of want to step back, take a little bit higher level view and walk through how we plan to stage the project as well as implement it, given the logistical considerations of working in the wilderness area. So as Pat mentioned, we're going to need a large volume of not only the rotenone substance, but also the detox agent, as well as a lot of equipment and personnel. So our proposal right now is to rely on helicopter transport for the actual rotenone detox and the detox equipment, primarily due to safety considerations about not wanting to risk a potential spill when transporting that along a trail using pack stock, but also from the weight considerations. All other equipment, the rafts, the personal gear, our food, the actual personnel conducting the treatment, we will be traveling and carrying that equipment with pack stock and backpacks. The North Fork project area is home to a lot of grizzly bears, and in order to minimize disturbance to those individuals, we've selected our flight paths to avoid prominent feeding areas during this time of the year, which include a lot of these higher elevation scree slopes around Red Mountain. So we'll limit our flights to the river corridor and avoid any sort of disturbance to these feeding locations. And we'll be able to conduct the flights in a maximum of 2 days to bring all that equipment in, and then a maximum of 2 days to remove it on the back end of the project. To minimize any sort of visible trammeling on the landscape itself, we are planning to use existing infrastructure, outfitter camps, cabins, as well as situating our spike camps in areas that frequently see camp use. So really, making sure we're not leaving a lasting footprint on this special area. As far as site preparation goes, we do have a large amount of beaver influence, particularly in the upper portions of the East Fork and the North Fork drainage. And as Pat was mentioning how rotenone works, to really get an effective kill in these streams, we want to make sure we have free-flowing water and not have all these impounded areas and backwater channels. It really presents a lot of challenges to effectively treating those locations. So a common method is to notch beaver dams. And we were doing a lot of reconnaissance over the last few years to identify the extent of beaver affected areas. And then last year, we actually did a trial investigation to see how notching would work in the project area. And so using hand tools, we determined that it was feasible to actually notch these dams. And we think we can do it on the scope of this project in the areas that require this. So ideally, we would go in, notch the dams within a treatment area, do our dye test, and then the next day treat through that area prior to beavers rebuilding the dams. And so you can see the notch is not very large. So if it's an active beaver dam, it's not going to be challenging for the beavers to come back in. So we're not going to have any lasting impact on the beaver population or the stream system itself. But you can see how effective notching was in lowering the channel or the stream back to its pre-impounded elevation and providing for a much more effective rotenone treatment through that section. So I put together a little quick schematic animation. Pat had mentioned drip stations, and just to give everyone an idea of how this treatment would actually progress through the drainage, overall, we plan to treat the East Fork and the North Fork separately, complete that, and then move over to the North Fork side. So we'd start at the upper portion of the drainage, and then work our way progressively downstream. And from day to day, we would be overlapping treatment areas within the main stem river section so that we don't have fish moving from untreated areas into the previously treated waters from the day before. So you can see how we're just basically creating a wall of toxicity moving down the drainage to achieve an effective fish kill. And then we'd be moving all of our gear and personnel over to the North Fork side, starting up there and then moving down to detox station. And as Pat mentioned, a key component of this whole plan is making sure that we have a minimum of 30-minute flow time above the falls. So we wouldn't have any toxic water going over the falls and impacting our current bull trout stronghold as well as the other fish species below the falls. So that's one of our primary safeguards with this implementation plan. As Pat had mentioned, the specific spacing of these drip stations will depend on what kind of water year we have in the year of treatment. So those dye tests would be critical to determining exactly how many drip stations we need in a given water body, as well as how far apart they will be spaced. Within the smaller tributaries, we'll be able to space them about an hour flow time apart. And then the main stem river sections and larger tributaries, anywhere from 2 to 5 hours apart. So you can see how all that information will really factor in to how many people we'll need on the landscape, how many drip stations, as well as ultimately how long it'll take to conduct the rotenone treatment itself. So if you're familiar with rotenone projects, you often see that folks are conducting 2, maybe 3 years of treatment to make sure that they've achieved complete eradication of the targeted species. With our proposal, we're planning to only do a single year treatment and then rely on genetic swamping to achieve our project objectives. You know, oftentimes if you do a really good first time treatment, you end up killing anywhere upwards of 95, 98% of the fish. So your subsequent years of the same amount of effort are yielding diminished returns. So given the logistic challenges, as well as not wanting to, impact this wilderness setting more than we have to, we think that it's a good compromise to do a single year treatment and then rely on genetic swamping to reach our project objectives. Particularly since, as I mentioned, the hybrids are actually in lower densities than what we're seeing in adjacent drainages. So we think with a one-time treatment, that'll maybe eradicate it in portions of the drainage and then significantly reduce it in some of the higher density areas. And then coming on the back end and stocking large amounts of pure cutthroat, it should reach our project objectives. So we're proposing 3 years of stocking. Multiple size classes would be stocked, not only to replace the lost recreational opportunity, but also so we have spawning very soon after project completion to further increase our chances of achieving the desired genetic swamping. So a lot of thought and planning went into identifying where we would get our source for refounding the project area. And ultimately the decision tree that was developed specifically for this project highlighted that our domestic strain at our Washoe hatchery, the MO12 strain, is the most suitable fish for achieving our project objectives. And 3 of the primary considerations, and I think probably first and foremost, was the ease of obtaining large numbers of fish. For this project to be effective, we need to make sure that we have the necessary amount of pure fish. So having that stable availability over multiple years is really critical to ensuring that we have the highest chance of success. And the good thing about the Washoe strain, although it is a domestic strain and we regularly use it for recreational opportunities, it was developed with conservation genetic principles in mind. So it was developed to do projects like this. So it retains a high degree of genetic variability. And in fact, a lot of the source populations that went into founding this brood source come from the South Fork, the flathead drainage, so just over the divide from this project area. So those fish are likely experiencing the same selective pressures, and we'd anticipate them to be highly successful within the North Fork project. And I'd just like to end on this slide. So this is coming from that Cherry Creek project that you've heard about tonight. And what I really want to highlight here is these post-treatment fish densities. So the closed circles with the confidence interval bars on them. You can see that following eradication of the hybrid species and the non-native brook trout, seeding the habitat with pure West Slope cutthroat trout, we see exponential growth following project completion. So really rapid recolonization of the habitat, and we're anticipating a very similar response within the North Fork project area. And I think it's also worth mentioning, this Cherry Creek project provides a lot of great foundational information for developing these types of projects. And they actually tested the MO12 strain against several different local donor stocks. And in the few years following seeding of that habitat, the MO12 strain actually had the highest survival. So again, I mean, that provides a lot of support that these fish will do well in the North Fork project area. And with that high survival, just further increases the odds that we'll have the desired genetic swamping effect to deal with any remaining hybrids that are still there after our rotenone application. And with that, I know that's a lot of information to digest. And right now I'd like to open it up to any questions. And again, following this question and answer period, we'll move into the actual public comment period. So again, here are the directions to.
Patrick, if I could jump in here, I think these are this is the old webinar ID and password, but the new one, that's okay. I forgot about that too, but the current info is on our website. And so it's right there below the video that you're watching. And if you do have a question, the raise your hand piece here is important. That way I can see that you've got a question, and so just press *9 on the phone, and then I will unmute you here. Sorry to interrupt you, Patrick. I'll turn it back over.
Oh, no. Yeah, I overlooked that.
So I'm going to give folks just a minute here in case we do have any questions. I see we have a few people on the phone, but I don't see any raised hands at the moment. So again, follow the webinar ID and password that's on the website rather than what's on this slide here. We'll just give it a few more minutes here and then we'll move on. We can always come back to a question if someone has one, so no big deal.
Say, Vivica, this is Pat Saffel. Could we put the dial and webinar ID information in a chat box somewhere?
That's a good question. Let me see here. I mean, it is on our website, so it's there right now, right below where the video is streaming. So it is there. Let me see if I can figure out a way to do that as well. I see we have a few more. Oh, I see a raised hand here. So we've got our first question.
Finally, yes. Hello.
Hi.
This was not easy to break in. Okay. My name is Sandra, and I live on the North Fork. My property borders about 4 miles of either side of the river, so I am a very... Oh, you can't hear me. Oh, I see a raised hand here. So we've got our first question.
We can hear you fine. I think what's happening is you might be screaming in the background.
It's repeating on the computer.
Yeah, just turn the volume down on your computer. But otherwise you're coming through loud and clear here. No problem.
Okay. All right. So I'm very impressed with all your science and numbers and all the work you've done. I've known about this since Ron Pierce talked to me about it early on. There are 3 things that concern me. First of all, you don't address a worst case scenario. I'm very downstream from the falls, but what's the worst case scenario? I read in the DEA that, and you address this, that you will not get 100% kill, so there'll still be hybrids or Yellowstone cutthroat or whatever in the system. And the restocking source, I don't know anything about. I don't know what an MO12 strain is, or there's an ease of obtaining it. It produces a large number of fish for 3 years. It's a domestic strain. You know, what's going to happen to the natural stuff downstream? And we already have too many bull trout that are eating all the cutthroat. What's going to happen down below the bridge at the at the North Fork Trailhead? What's going to happen?
Well, thanks for those questions, Sandra. I think I'll start off with talking about the donor source, and then I think I'll turn it over to Pat and Don to talk about the question about worst case scenario, and they can shed some light on the other multitude of projects that have been completed around the state. But as far as the MO12,
well, I mean, that's fine that they've been completed and they've been successful, but you know, there's always a worst case scenario. So I'm just wondering what the plans are for this one.
Yeah, no, that's a great question, and I think Don and Pat will have a lot of good information about, you know, all of our contingencies as well as kind of built-in safeguards. As far as the actual donor source, the MO12 strain that I put up, I mean, that's just basically fish jargon for what we call our West Slope cutthroat trout that we've developed at our Washoe Park hatchery. And right now it's got about 16 different populations that have contributed to developing it. You know, from the South Fork Flathead, the Clark Fork, the Bitterroot. So maintaining that high genetic variability that makes it a successful fish for conservation projects such as these.
But they're not technically native.
No, they're native. I mean, I think, you know, the genetics world always gets a little gray as to, you know, what really constitutes, you know, a native fish. I mean, do we go down to the level of individual alleles, or is it enough that it's a pure West Slope cutthroat trout? Really, the only real difference is, you know, ultimately what selective pressures contributed to that genetic strain. And quite frankly, the fact that in the Blackfoot, a lot of our pure fish that might have been suitable to refound this project area either have disease concerns or had a slight level of hybridization. So one of the only high contenders that came out of all the baseline sampling throughout the Blackfoot to identify a donor stock was in the middle part of Nevada Creek up Cottonwood Creek. And I mean, I would argue that the MO12 strain has more in common with fish in the upper North Fork than a fish in an isolated population in middle Cottonwood or middle Nevada Creek drainage. You know, the South Fork, the Flathead drainage, where a number of the donor populations came from, or, you know, a hop, skip, and a jump from this project area. So very similar evolution processes and a very appropriate fish. I mean, yeah, it's true. We didn't, we're not proposing to develop a localized strain, but I think from what we saw at Cherry Creek, this MO12 strain is very successful in refounding the habitat, and the fact that it had very high survival, that just further increases our chances that genetic swamping will have the desired outcome. And
The
I mean,
desired outcome of what? An MO12 or a true? I mean, there's no way to get back to a true West Slope cutthroat up there, probably. I mean, the true one.
I mean, the MO12 is a true West Slope cutthroat trout. I think ultimately what we'll end up with is probably pockets of 100% pure West Slope cutthroat trout, and then those areas that currently have the highest density of hybrids and might not have 100% fish kill, we might have anywhere from 90 to 99% genetic purity. So still it would look and behave like a cutthroat, but you'd have to take a fin clip to tell that it's not 100% pure.
Well, yeah. You're trying to replicate something that really cannot be replicated, but you're doing it as best as you can, is what I take away from this. I mean, you can't kill all the fish you need to kill because of the logistics up there. Sorry, I have an age-old voice that's blocking out. So you can't really restore the original West Slope cutthroat. I don't think. I'm just trying to get sort of a picture of, I know the years that have been put into this, and I know how hard it is to access where you're going. I've been up to the falls. But I just, I get a little tired of collaring everything, banding everything, and trying to, redo what God has done to us for whatever reason, and what people have done to us. And I just worry. I've spent a lot of time and effort protecting my approximately 4 miles of the North Fork from cattle, putting in fish screens, et cetera, not overfishing, which is currently happening. And I'm just trying to protect the fish that are here. And if they're funky fish, and they can live here, that's fine. I'm just not so sure we have to overmanage everything. So that's kind of my standpoint of where I come from. And I think you've, other than the worst case scenario, and what happens what do you think happens if you don't kill enough fish that you're trying to kill? What's the ramification of that? Are you going to go back and do the same?
At this point in time, we're just proposing that one-time treatment, and we plan to start stocking that same year, so in the fall, right after treatment. So our intention now is to do the best, most effective rotenone application possible, and then just commit to this stocking of pure fish. Luckily, we did the bioassay in 2018, so we feel pretty confident that our rotenone concentrations are going to be dialed in and be effective without having to, say, do a 1-year treatment and learn from those mistakes and then come back in subsequent years. So I think we learned a whole lot from that 2018 bioassay about the challenges and what is necessary to be effective up here. So I think with that information, we can feel pretty confident with this proposal of just doing the 1-year treatment and then our genetic swamping for the 3 years following.
Yep. Okay. How many people tuned into this? Do you know?
I don't have a way of seeing that at the moment.
Yeah.
I don't either, but I might be able to get info on that. And let everybody
is
know.
anybody else asking a question? I guess nobody.
Not
else
at the
cares.
moment, but I did want to ask for that, but I also wanted to point out, thank you, Patrick, for updating the info here. So now the correct webinar ID and password's right here in front of you too. So that is correct. Oh, we do have another raised hand. Are we okay to move? And we
Yeah, that's
Actually,
fine.
well, Sandra, I definitely want to get to your question about worst case scenario, because I'm sure a lot of other people are might be wondering the same thing. So I wonder, Pat and Don, would you mind taking a stab at that one?
Yeah, Don, do you want to say something or you want me to go?
Go ahead, Pat.
Okay. So yeah, Sandra,
So which Pat is this?
this is Pat Clancy.
Okay.
I'm the retired guy that has done a number of these projects in the past, but I get hired back by the department through a temporary agency. I'm a Kelly girl.
Okay.
So to help on these types of projects. So worst case scenario, are you thinking about, if the rotenone comes over the waterfall, how far downstream would it kill fish, or what would it,
Yeah,
might it kill
yeah.
beyond our target?
Right.
Okay. Well, in my experience, we're going to set these neutralization stations up at least a half an hour of stream flow time above the waterfall. And I've never been involved in a project where we could not neutralize the rotenone in less than half an hour.
Okay.
So we, based on our experience so far, that's been an achievable goal.
Right. And the barrier of the waterfall, the purpose of that is to what?
Well, the waterfall prevents
Bull trout from getting upstream.
Yeah, or any non-native fish as well. Primarily, it's, we want those barriers on these types of projects, whether it's a natural waterfall or a man-made structure of some sort. We want that prevents non-native fish from reinvading the area after we've completely gone through.
Okay.
And then I mentioned during the presentation that we have to have 2 independent operable neutralization systems. I don't know if you heard that part, but our Fish, Wildlife, and Parks policy requires that there be 2 completely independent neutralization systems on site and set up and ready to go. I mean, if one of them were to just suddenly stop, we could just turn the other one on without losing more than just a couple of minutes. And that
protects your 30 minutes.
Yeah, that maintains the neutralizing in that 30-minute. And then additionally, we have to have twice as much of the neutralizing agent on hand as we expect to use. So, if something happens while we're up there doing a project, that we end up having a rainfall event that really increases the stream flow while we're neutralizing, and then requires us to use more neutralizing agent, we have a pretty substantial amount still on hand to get through the neutralizing period.
Right, via helicopter or whatever.
Well, it will be on site up there. I mean, it'll be right there.
I mean, delivered via helicopter. Or
oh yeah, it will. Yeah, prior to any
rotenone
rotenone going.
Whatever. Yeah.
Right, prior to any rotenone being applied to the water anywhere in the drainage on any of these projects, these neutralization systems have to be ready to operate.
Okay. Well, that's good to know. Well, just know that there are not very many of us who have the blessing to live on the North Fork farther downstream, but we care a lot about it. And maybe not everybody was on this call, but we work very hard to protect the fish, try to protect them from overfishing. Oh my God, it's been a zoo. And things are changing. The bull trout are in charge, and the cutthroat are frightened. So it's kind of an interesting change in 27 years. Anyway, I care a lot about it, and I appreciate you answering my questions, and I hope it all works.
Sandra, this is Don Scar. I'll add one thing to what Pat Clancy said, and that is, per requirements on the label, when we have the rotenone delivered to the site, So we'll have barrels of rotenone there. Those will be stored in a place where, you know, if there was a spill, they aren't going to get into the water. And then for any given day that we apply rotenone, there's requirements on the label where you have to set up a secondary containment system when you transfer rotenone from barrels to the vessels that people use for the day, take out to the stream. And so in any given day, there will not be that much rotenone that could even get into the water, even if an accident occurred. So that's another precaution that'll be taking place.
Okay. Thank you. All
And
right.
Sandra,
Well, thanks for talking to me.
Sandra, this is Pat Clancy again. I just wanted to say that I think all the agency people involved in this have a full, an amazing amount of admiration for the Blackfoot landowners, because we know you guys have done a tremendous job up there.
Well, good, because we have. And we just care a lot about it. So thanks for listening. Okay.
Thank you. And I just wanted to say thank you for saying your name and where you were from. I forgot to remind folks of that. But you did that on your own. And so this is my reminder to future callers to do that too. So we make sure we have that. So I am
good.
Yeah. Thanks for leading the way.
Okay. Goodbye.
Bye. So we have another caller here with a question, and just a reminder too, we're still in the question period. We are going to move to comments next, but just a quick reminder on that. Okay.
Hi there, this is Clayton. This is Clayton Elliott, Conservation Director with Montana Trout Unlimited. I actually was a little bit confused. I had more of a public comment than a question, so I guess I'll wait.
Dave Vivica, this is Pat Saffel. I think Sandra gave quite a bit of comment during her question and answer. I think we're mixing it, and that's fine. I think Sandra was going to sign off, but we'll just deal with it.
Great. Well, I'm happy to quickly go now. Just again, Clayton Elliott, Conservation Director for Montana Trout Unlimited. Thank you so much for the opportunity to comment tonight and the great presentation. Just wanted to thank the agency for the hard work, good science that they put into this project and proposal. We enthusiastically support the proposal in front of the public today. Submitted written comments, and I'll leave most of those, but I think the science is strong, and just wanted to go on record saying we certainly support this and think that this is a great way to move forward with the conservation objectives for native fish conservation. Really see this as a future model for other conservation areas and this work that we continue to do to preserve native species. And, you know, certainly from Montana Trout Unlimited, wanting to speak on behalf of our various chapters in the area, many of which you have interests and concerns related directly to West Slope cutthroat and native fish conservation and bull trout. I never thought I'd hear there were too many bull trout, but, Pat Barnes and Helena, Pat Barnes to you and Helena, big Blackfoot chapter in the Blackfoot Valley, West Slope chapter in Missoula, and the Flathead Valley chapter as well. Just wanted to raise their level of support for this as well. So thank you so much, and thanks for the good work. More detailed comments in writing that we've already sent your way. Thanks.
Great, thank you. I don't see anybody else at the moment, but I'm gonna pause here just a minute to see if we do have anybody else raise their hand. And again, all the reminders are up here now too, and all that info is right there on the page where you're watching this, but the comment deadline has been extended by a week. So now through August 14th instead of the 7th. We've already had comments coming in. So tonight's meeting is by no means the only opportunity to make those comments. There's anytime you can log into the website there and make your comment or send your email or mail a comment in. So lots of different ways to do that.
And I'm going to go ahead and add my contact info to this slide. So, I mean, if anybody, either doesn't feel comfortable asking questions right now or thinks of some tomorrow or over the course of the next 2 weeks, feel free to give me a call, and I'd be happy to chat and address any other concerns you might have.
And I haven't seen anybody else join us, so I think we may give one last call here for any outstanding questions or comments.
Alec has his hand raised.
I'm getting there.
There we go. Can you guys
well,
hear me?
Yes.
Thank you. Thanks for catching that, Pat. And thank you, Patrick and the other Pat, for great presentation. I actually missed a middle chunk of that when my computer died. So Alec Underwood with the Montana Wildlife Federation, much like Clayton there, we will be submitting some more detailed comments, but just, wanted to again, like Clayton, articulate that we appreciated the presentation here, and we see this project as having immense value for native species conservation. So again, just to appreciate the department's efforts, and we'll be sending some official comments soon. So thank you very much.
Great, thank you. There are a few other attendees on here, so I'm just pausing a minute to make sure nobody else is going to raise their hand, or that I'm not going to miss it again. Okay, well, I think that may be it in the way of comments and questions. The info is up here on the deadline and the how to comment after this meeting. And Patrick's contact info is on here too for questions. Patrick, did you have, or anyone else have any closing words?
No, I definitely want to thank everybody that tuned in tonight, and a lot of great questions and comments, and certainly appreciate getting all the comments, both positive and some negative, that we have received in the comment period thus far. And I think all of this lends to developing a better project on the ground. And I think everybody that tuned in cares about fish in one way or another. So I think we're all just working towards achieving, our fisheries management and conservation goals. And I think tonight was very productive. And once again, yeah, thanks for being flexible with the reschedule. And I know we went through a lot of information. So like I said, don't hesitate to reach out to me or any of the group. And we're more than happy to be as accommodating as possible over the next couple of weeks, and address any further questions or talk at length about any components associated with this proposal.
And we'll work to get the recording of this meeting, including the presentation, up on the website just as soon as we can. So look for that later this week too. All right, thanks everybody.